here with us, Jeff. Jeff was one of my better grad students, and he stands out in many different ways from a lot of the other people. Actually, I should have had you guys read his paper. He published a paper in my lab looking at insect viruses and how insects respond to them on a transcriptomic level. So we talked about microarrays already at one point. And so he was really instrumental in getting a publication out that was probably particularly unique and resulted in some pretty nice collaborations with some pretty famous, at least in that field, famous people. What was some of the names that we ended up having on the publication, Jeff?
Heiko Vogel was on there. So obviously Max Planck Institute out in Germany. Holly Popham, who used to be at the USDA, and now she's at some ag biotech company now. That's she's one of the lead or on the board now for that company, and she has several publications to her name, as well as Jonathan Breitenbach, who still, I believe, is with the USDA there as well.
We have Dr. Meager joining us too, Sean. Jeff. Did you ever take a class with Dr. Meager?
I've taken several classes with him. One of the hardest professors I've ever had.
Even with going to University of Kentucky and stuff.
Yes. Yeah, absolutely.
Oh, wow. Yeah, I was saying that Jeff had actually published a paper, and that was one of the standouts. A lot of people that do get publications in my lab, it's usually years later, and that's when I finally got around to writing it. But we were able to get a publication out really rapidly when Jeff was here. So, and again, that was with insect viruses, looking at how the epithelium of the caterpillar's midgut deals with viruses. You can't hear Sean, is that correct? Can anybody else hear?
Yeah, I can hear you. You might have it muted. I don't know, you may have to check your audio settings.
Hey, check your audio settings.
He can't hear us. Tell him that.
You might have to log in. Isn't it like you have to push the audio button?
Yeah, but he can't hear us. Tell him that.
I'm typing now. I guess I should spell here correctly. But everybody else can hear, right?
Yes.
Is there anything I can do? I think you have to dial in, right?
You might have to.
Hear you now.
Go. Well, I won't take any much. Let me just move this out of the way. And I'm going to pass over the torch to Jeff. But thanks again for giving a seminar for our students. And, like I said, I really appreciate everything you've done. And it's one of those where I think we developed over every generation of students, you're one of that. I developed a pretty close relationship for your time here. So
sure, absolutely.
Let me go ahead and make you the host.
Okay.
So now you're welcome to put your PowerPoint up. And
all right, let's go ahead and share one of my screens here. All right, share that. All righty. Everyone can see my screen okay, correct?
Yep.
All righty. So again, I'd like to definitely thank Dr. Musser for inviting me to give the talk. My presentation is really going to be about taking some of my background and how I've applied it from a strictly academic and R&D background into an industry career for science, and what I've done actually extending some of the skills that I've learned in entomology and molecular biology, pushing it over into a completely unrelated field, somewhat in fermentation science and bioconversions. So my talk title is Bioconversions, Translating Academic Skills for an Industry Career. So just to go through a little bit more of an in-depth background on me, I have an undergrad in microbiology from Western Illinois University, as well as a master's degree. some of the things that I worked on in my undergrad that related directly to the work that I'm doing now, worked on some of the fermentation pathways for soil isolates of Bacillus species, particularly Bacillus thuringiensis, and isolating a soil isolate actually right outside the door at Wagner Hall that was able to outproduce in alpha amylase production compared to most of my cohort in my class, which was pretty cool. And then also working on some biotech aspects of cloning genes out of leuconostoc species, putting it into E. coli for high throughput expression or increased expression in a different novel host system. While doing my undergrad, I started looking into grad school. I was fortunate enough to have Dr. Musser sponsor me and bring me onto his lab. Wanted to keep that microbiology emphasis to some of the career aspects that I had. And so he had mentioned that at one point he had an insect virus that I could work with. And so it was really intriguing to me. The type of virus I worked with was called a baculovirus. Natural host range is usually relatively species specific, but mostly in lepidopteran species or caterpillar species. And I, like Dr. Musser had mentioned, I was able to get a publication out of it relatively close to the same time that I officially graduated from Western. So the title of that publication was Gut Transcription in Helicoverpacea is Dynamically Altered in Response to Baculovirus Infection. So again, 24 hours post-infection, looked at the transcriptome of the gut tissue, see how it responded. Pretty well disrupted in terms of how it typically functions when it's not infected. So most of the digestive genes as well as major metabolic pathways are dysregulated, so the gene expression had decreased significantly over time, especially in that first 24 hours. There were genes associated with the immune response that is stimulated, so seeing pretty canonical, pretty much stomach flu kind of response in caterpillars. So that work landed me at the University of Kentucky, where I ended up finishing with a second master's degree. Originally, I was going for a PhD, but it was interesting. My wife and I had twins right in the middle of my career there. And I had made a decision when I had passed my qualifying exam that I was going to step down with a second master's degree. Mainly because the $18,000 a year stipend at the University of Kentucky wasn't going to be nearly enough to help support my family. So that's a decision I would make 1,000 times in 1,000 lifetimes to support my family. So I put my academic aspirations on hold and finished with a second master's that was really diverse in terms of my education. While at the University of Kentucky, being in the more molecular biology, biotechnology-based group in entomology, half of my academic training classroom-wise was in the College of Medicine. I also took several classes in chemical engineering as well as ecotoxicology. So very diverse background. Finished a publication, or finished with 5 publications actually, all based on risk assessment profiling for transgenic crops and their purified incorporated protectants. So that's what my academic background is. Translating into, obviously, the reason why I left from grad school is to support these two little guys. Obviously, here on the left, you can see what they were when they were maybe just under 2 years old, and then over here on the right was just this past Halloween. So they're almost 5 years old now. I can't even believe I've made it this far as well as them. So going into more of my industry training, right at the time when my wife was almost done with her pregnancy, I started interviewing at a company called ADM, or the Archer Daniels Midland Company. It's a company that's actually in Illinois, Decatur, Illinois specifically. They have several manufacturing facilities throughout the world, as well as throughout the United States. That particular job was the one that took and translated me from not only understanding the difference between academic R&D, but taking that focus and shifting it into R&D for a purpose of a company's benefit. So understanding how to not only take what I've learned, unlearning it, and then relearning it in a different facet was one of the biggest things that I had to do while I was at ADM. And I had a fantastic mentor there. His name was Bill Blaylock. He was key in basically what he said. It was making Jeffrey 2.0, which was really what he did. So yeah, that work entailed a bunch of different things, from not only looking at engineering bacterial strains for fermentation of amino acids, but also specialty proteins, enzymes, and everything you can think of coming into the bioethanol field. So several different things there. Produced several different strains, of which several had scaled to commercialization within roughly about a year or two of me being there. Several different bacteria, as well as yeast, were the main focus of what we were looking at. And then also provided some plant support for the different endeavors of our facilities that we had at the company. From there, I transitioned to a company called Corteva Agroscience. This is the old Dow AgroSciences. That was the spin-off company that came out of Dow DuPont after that merger in 2015. At this time, this was a contract position, still one I definitely appreciate. This has taken my bioengineering skills and translated into more of the bioprocessing side, so truly understanding how an organism responds in a bioprocess, be that fermentation or for the production of certain commodity chemistries. Particularly here, I managed the external strain engineering projects that we had, particularly with strain engineering companies. Those strains would come to me at Dow Agro or Corteva Agroscience, and I would put them through our process, which would be to ferment different insecticides or fungicides as well. Worked at two different particular scales, a 2-liter and a 30-liter scale. And that work would eventually translate and scale up to manufacturing. So I felt like I had earned the title of Grim Reaper for a lot of external strain engineering strains that had come into our facility, mainly because most of those strains would come into my hands and die, unfortunately. It's just kind of the role that you get with it, unfortunately. But at the same time, it proves the stringency of what we expect in a process and what we expect to make it to manufacturing scale. In terms of the most recent experience I have was at a company called the Andersons, particularly one of their ethanol plants. This is where I was a quality assurance, quality control lab manager, as well as managing the fermentation process itself. Here I was able to really expand and develop myself into really what I expect in terms of understanding fermentation management and bioprocessing. Here, one of my direct reports, the laboratory lead, is actually another former grad student and undergrad for Dr. Musser, Jason Tudor. He has now backfilled me in my position there, so he is now the lab manager at this company. But for a little bit more understanding for what this is, I served as an interim production manager on 2 different occasions when we were looking for replacements for that particular position. I was the fermentation subject matter expert, as well as a site leader for our 5S and 6 Sigma programs, both of which ended up producing quite a bit of optimizations at the plant. Specifically, in 2 years, we added 23 million gallons worth of production, which in terms of a manufacturing facility, gallons out the door is really what pays the bills. And so 2 of the projects I had for my Six Sigma Greenbelt ended up being roughly $12.1 million in revenue increase for the physical facility itself. At the time, I was looking for jobs, trying to further my career and advance myself. I had interviewed with this next company, AlphaZyme, who is in Jupiter, Florida. They are a company that ferments medically relevant enzymes, particularly some that are associated with COVID-19, which is really interesting. With this particular job interview, I would have been more than happy to go for the opportunity, but considering everything going on with my family and making sure that my wife was still happy, I decided to pass on this opportunity, but at the same time, made a good impression with them and was able to start with them as a bioprocessing and fermentation consultant. So still working with them today, mainly going through and helping them optimize their system for what they have, automate the lab, better understand the dynamics of what fermentation is for what they're trying to do, and then help onboard new strains and new engineering traits for what they need to do and help them keep producing enzymes that are part of their contract agreements. This basically is an offshoot of the skills that I've learned throughout my entire industry and academic career. So this is still a nice little project that I get to do on the side, even though I didn't join them full time. And where I'm at currently, I'm now a principal scientist in fermentation and bioconversions for the Kraft Heinz company. That is the mac and cheese and ketchup company. So I am now based out of Chicago, Illinois. I lead the fermentation technology platform within our tech discovery and development group. I serve as a lead for some of our natural antimicrobial platforms, as well as I self-promoted leader for our bioreactors that we have within the group too. So this position really deals with novel fermentations, not only for flavors or colors or anything you can think of related to food-based ingredients, but also for natural preservatives and antimicrobials that way. Work directly with our pilot plant that is attached to our facility, and we help scale that up as well. Providing some plant support for some of our external or internal company facilities that are located throughout the country, most recently working with a group in Texas as well as in Champaign, Illinois. And then some of my role deals with microbial physiology, optimizing some of our strategies, both internally and externally, for engineering strains for the company. So I wanted to ensure that giving everyone just a diatribe about my background was not the only thing that I did for this presentation. I want to make sure I did a case study for everyone so they understand necessarily what's kind of required of people in various level positions once you get into industry as a scientist. So this is the work that I did at the Andersons, which as I mentioned, was the plant that I put on 23 million gallons worth of production on in a two-year span. Essentially, my first day on my job, I wanted to figure out what the company really needed, and particularly that plant. And the quote that I got from the plant manager at the time is, what we need you to do is focus on fermentation and bring that as a first consideration for the facility. It's something that they wanted to push as a focus, not only for that particular plant, but our other plants that we have as well. So it was really key in trying to make that more of a stable process and understanding how specifically we can control a relatively variable process, depending on not only sterility, but as well as opportunities to enhance what we can produce. And so some of the work that I ended up doing was to really push to redesign the fermentation recipe, specifically making sure that it is as consistent as possible. Despite increasing the amount of ingredients going into a fermenter, we should be able to better predict what we get out on the other end. Not only that, I implemented a increased sampling regimen to have better eyes on fermentation so we can catch problem fermentations a little bit more quickly, as well as going through and doing some cost-saving efforts, not only in terms of reduced failures of fermentation, but also reducing our antibiotic usage, providing better nitrogen sources, as well as overall increase our production by getting better ethanol titers at the end of fermentation as well. So this is the facility in particular over here on the left. This is just a Google Earth image, but this is the entire site layout. You can see up here towards the top of the image with those couple roads coming in, these first 10 tanks that you see right here, the nine of them, except for the one on the bottom left, are all fermentation vessels. These are 807,000 gallon fermenters filled to about 96% full every single time. It's a heavy enzyme usage type of fermentation. We basically take ground corn and water, add enzymes to it, and ferment that by converting the liberated dextrins into glucose for yeast to ferment into ethanol. While that seems like a relatively rudimentary process and description, it can be very dynamic in that there are several different types of bacterial species that come in at any time. There are nitrogen deficiencies, not only in the crop health, depending on harvest year, but also different nutrients that fluctuate throughout the year based on not only proximity to harvest season, but also when you get into the last remnants of your previous harvest season when everything starts to collapse in terms of stability. So like I had mentioned, the main concerns for certain fermentation systems like this are contamination events, or what the industry calls as infections, relatively low ethanol titers, as well as a low plant chemistry pH. So running under a pH of about 5 tends to be a little bit more harsh on your system, depending on what strains you're using, as well as what enzymes you're using, as well as being able to try to run newly engineered strains in our facility, which typically do not respond to some of those highly dynamic systems in terms of altering conditions. And so the ethanol overview in terms of what this is, basically we get inbound corn that we grind up, which goes from our grain storage to our hammer mills. That's the grinding system. Goes into a slurrying process, which is initially hot water and corn flour, which then in that mashing process starts to gelatinize and pull all the long chain starches out of the corn, which then gets hit with alpha amylase, breaking those those polysaccharides down into dextrins, so shorter chain sugars, which then goes through another milling process, a wet mill, if you will, where it goes through basically a disc mill, grinding the corn together to get better shear, better contact with your enzyme as well, and then goes into a long resonance liquefaction process, which then goes right into our ethanol fermentation tanks or our yeast prop tanks. That roughly takes, depending on your facility size, anywhere from, I would say, probably about 45 hours, depending on how large your facility is, all the way up to 75, maybe even 80 hours, depending. So anywhere from 2 to 3 days. From there, you go through a separation system, essentially, in downstream processing, where you take the solids out of the beer that you ferment, as well as the dewatering process through centrifugation, and then you distill off your ethanol. So that's essentially what the downstream processing looks like. Once you go through distillation, you have 95% alcohol, or roughly 190 proof, which then goes through a dehydration process through the molecular sieve, and eventually turns into 200 proof, or 100% ethanol, which then gets a denaturant, which depending on your facility, it could be isopropyl alcohol or a bittering agent called Bitrex, or natural gasoline if you're in the fuel ethanol world, which is what this is specifically on. And then the solids part of your downstream processing is called whole stillage, which then goes through the centrifuges to specifically isolate the solids and liquids, which then get reapplied and recombined later on in our grain dryers, which then gets sold as a product called distillers dried grains. And that goes into animal feed markets, particularly livestock. Some of the evaporants go into our evaporator system, which then we can strip oil out for, so crude corn oil extraction, which then can go into different industries. Animal feed is a supplement where it can be used, as well as going into serving as a feedstock for biodiesel production or renewable diesel even. And so for that process, again, I'm trying to make sure what I can do to increase the stability of the process. And so that's really what my focus was over the last 2 years of my career at the Andersons, is how do I make fermentation more predictable, as well as how do I optimize the process. And so what that means is a bunch of different graphs I'm going to show you guys, particularly running different types of strains in the process that are all commercially available through different vendors, as well as understanding some of the plights that our facility had with different optimizations that we had, particularly something called a distributed control systems update, or basically a computer update. And the upsets that that can have on your facility. We use 3 different strains in this process, something called Transform Yield Plus, Transform RB3, and Thrive GX, which are all commercially available by companies like Noland Biofuels and Distilled Spirits, or DuPont. And then we also use another strain called Ethanol Red. These are all commercially viable strains, not only in the ethanol field for fuel, but also in other distilled spirits fields as well. So this is a very large graph that focuses on the last 2 years of the work that I did. So from December 2018 all the way through this last little bit, which is November 2020, which is when my data cut off. There are different fields in this area where we look at either running different strains, which this DuPont strain is the red diamonds over here on the left side in December, transitioning to a single strain, Ethanol Red, which are the black dots through most of the year in 2019 and 2020, as well as certain events like the DCS migration that we had from September to November in 2019. Another strain trial we had late November through December of 2019, and then also optimizations that we made to the fermentation process over here in 2020. Now, as you can see, there's a lot of variability in this process. So each one of these are focusing on our ethanol titers at the end of fermentation. Specifically, we're running anywhere from 13 to 15%, which is what industry standard is. Where you try and diversify yourself or make yourself better is how consistent that process can be. What you want to prevent are massive drops-offs in your ethanol production, which, as you can see, there are some different events that can be controlled and cannot be controlled. Those are things like contamination events, which can be operations related, but they can also be, natural kind of events too. So if you have a power outage, you have to get everything back up and running. You might lose time on cleaning aspects, but you also might not get the heat resonance to kill bacteria in a pasteurization process that you would have before, because you're starting up from cold. Right. So there are several events where in the summer, if you have thunderstorms, most of these facilities are basically on electrical grids in the middle of cornfields and soybean fields across, the United States. So the infrastructure for how well these facilities internally can handle issues are fine, but it's usually your utility inputs that don't necessarily have the greatest stability. So those are just different types of things that you have to encounter in this kind of a field, but your ways of handling those response elements to how quickly your plant can go down but come back up, how quickly you can get the process moving forward again, all impacts everything that you see here on this graph. Particularly, the one part I really want to emphasize on is after I started really controlling the process by increasing sampling here late July into August of 2020, you can see those 2 red bars shrink for the rest of the year. Those are what you call the upper and lower control limits for what this thing is, and this is called a control chart, which shows you your range of variability across time, which is really nice to see when you really hone in and control your process, how you can reduce your variation, which makes the process more predictable. And so that's what our main focus was on this latter half of 2020, is how can we make this not only a better process, but a more consistent process. And so this is just a NOVA table looking at all the months, all the production months that we had over the first 2 years. Essentially what you're seeing is the diamond in each of these sets of months really is the actual average, and then your two blue lines that flank top and bottom, technically your upper and lower control limits, but that's your range of variability. And what you see here is when you have over here on the right, we have something called your post-hoc analysis, which for these, it's easier to show on each pair of students t-test the difference between specific months. But in an ANOVA data, what you're going to look at is another post-hoc analysis called a Tukey's HSD, which is the Tukey's honestly significant difference. Very robust tests to show you specifically, is there a difference between the variability of your months, but also the variability of your data across a range of time. So this truly supports what you see here. This shows you horizontally the data. This shows you the data vertically. Essentially is the main difference here. So you can even see it's very reflective of the work that we've done. Again, focusing on August 2020 through November 2020, the tighter variation and spread on our range of data really shrunk down in this time frame. And also you can see that in those months, our titers had steadily increased over time. So another part to this data is looking at our contamination events. And so this really was a struggle for the facility as a whole, whether it was dealing with not only cleaning, but also different types of antibiotics we had in the process. And so for the first year, we were just trying to control our contamination events, which we ended up finding out we had some resistant populations in our cook process, which made roughly every 3 weeks we were getting infection spikes. So what we ended up doing is we had alternated our cleaning that we have done, as well as focused more on reducing the number of antibiotics we were using in our process for something that's more potent. And you can see here in 2020, what we had focused on was right at April, the month of April. We had our COVID shutdown, and so we cleaned out the process as best as we could, making sure that every single pipe and surface was as clean as possible, and then focused in on getting more strong antibiotics that were used in tandem more consistently to reduce the number of bacterial populations that can become resistant. Now, what you see, unfortunately, in July is some issues in the process that were operator related, which contamination events happen if certain vessels aren't cleaned well enough, or if you have an issue where you're trying to get, like I said, the plant started up again, which is what these were associated with. And so a couple vessels did not get cleaned as well as they could have. And so we had an outbreak, and particularly in the field, lactic acid tends to be very lethal towards yeast. And so lactic acid bacteria are primary concern, which is why we use the antibiotics we do. Again, in the month of August, which is where our variation decreases most significantly throughout the year, what we had ended up doing was increasing fermentation sampling by adding another sample in our seed train, which is our yeast propagation process, as well as adding 33% more sampling across fermentation. And what we see is a decrease in overall variation to a point in November where it was the lowest it ever has been in our plant's history. As well as, even though we have these random spikes every now and then, roughly, every 20 or 30 firms, we did not lose a single one of those fermentations. And so reducing contamination increases our output by not having to blend off a fermenter, as an example, losing roughly about $200,000 every single time we had to. So the reason why I'm showing this as a particular example is because these are the kind of things that are asked of you when you get into the industry. Your managers are going to say, we need you to fix this problem. How are you going to do it? Sometimes it takes 1 or 2 changes. Sometimes it takes 2 years. So this was a definite achievement for our facility, and definitely proud of this work, mainly because, coming into the industry with one of the plants that ran relatively inconsistently to be leaving the industry, knowing that your plant is one of the premier leaders in the field, is really the kind of impact you want to make. And so that is definitely acknowledged in the ethanol field for sure. So this is, again, just going over those particular phases, like I had mentioned, 33% increase in fermentation, as well as implementing a new strain. The new strain that we had put in produces something called methylformate, which ends up being a little bit bactericidal itself. So once that strain got in and on its own recycle, you can see a decrease in bacteria content as well. So I put this in because I wanted to relate the science to the process as well as making money. So again, what kills yeast during an ethanol fermentation, right? It's not only environmental stressors, but it can be things like lactic or acetic acid bacteria. So they ferment organic acids, which tend to be synergistic. Lactic and acetic acid tend to kill yeast very efficiently. And so what happens is you go through a pH change on the intracellular side, and you end up sending a DNA fragmentation signal, which causes the cells to die through an apoptosis-like cascade. So I remember, as an anecdote, I remember the first time I ever heard of apoptosis was actually in Dr. Meager's parasitology class. And so that's kind of why I put this in, because I still remember these things from my undergrad. And so it's nice to kind of have that throwback every now and then to make sure that you're still up to date with what you're talking about too. So really cool stuff. Again, this is just proving what some of those contamination events can do to yeast, which are very fragile in this kind of process, despite them being robust as they can be. They're facing a ton of different things in a plant at the same time. So this is really, really critical for our operations. So again, flipping from reducing contamination to increasing ethanol titer, you can see in that same time frame when we focused on making sure fermentation was staying relatively clean and consistent. You can see variation from August to September to October, and even into November, decreased every single month, and ethanol titers increased over that same time frame too. So those are really some of the key events that we wanted to focus on and make sure that we're continuing to push through the plant. And again, those were optimizing the recipes as well as in putting in the new strains. And so this is just the statistical data that shows the same thing. Again, using ANOVA and t-test data to prove this out, you can see in November, the tightest variation on our spread of data, as well as the highest diamond across all of these other different months, shows that this is one of our best production months in our plant and that it's had in its history. So this is a phenomenal move. So kind of circling back to the purpose of talking with the class is to ensure that we kind of go over what it takes to be ready for an industry career as a scientist. The one thing I will definitely say first and foremost is adaptability. You're always going to be pushed to do new things, whether it's within your comfort zone or not. And I was told, one of the first things I was told when I got into an industry setting is don't fall in love with projects like you kind of do in grad school or your undergrad, because corporate will tell you to drop it if they don't see any value in it. So keeping that focus on, you could put 2 years of work or 3 years of work or 5 years of work into something, and company says, well, this is great, but we need to focus here now. And so being adaptable is, don't let your feelings get hurt because of that. Being proactive versus being reactive. So being ahead of the curve when it comes to being on the leading edge of technology will help you in the long run, not only for things like publications, which in industry, you're not really focused on publishing in a journal, you're more focused on publishing patents. And that lends itself to being on the forefront because you want to be ahead of your competition at all times. But also learning how to extend your boundaries to, as an example, if you focus on it like a campsite, right? It's very dark out. You have a campfire. If you make a campfire bigger, you can see more of the unknown. And that's what it's like to be on the forefront of being on the leading edge of technology. And so you always want to be thinking ahead rather than, okay, our competition did it, now we need to do it. So you have to be ahead of them. So if you can see what the market wants before the market needs it, that's how you get in on that precipice of being on the cutting edge. And then this is something that I definitely had to learn too, not only in academia, but also in industry, is knowing when it's time to move on. Some companies are definitely going to see your value. There are times where a company will not see your value for what it is. There's no hard feelings in it. A company is going to do what it needs to do to be self-sufficient. And if you're not part of that plan, it's no fault on you and no fault on them. But you need to be able to identify that yourself and be able to take the step, while it could be uncomfortable, to move on in your career, or maybe even take a change in your career too. When I was at the University of Kentucky, going into industry was not the first thing I had thought of. I thought I was going to be kind of like Dr. Musser or Dr. Meager, go to a university and be a professor. Things didn't work out that way, so I had to know when it was my time to actually change what my focus was and push on to a different field. So it was definitely rewarding, definitely something that, if I can go back and do it again, I don't know if I would make any changes. I love what I do. Fermentation is one of the coolest things I've ever done in my life. And it's definitely something that I find a lot of value and a lot of rewards in. And circling back on that is making sure you have self-confidence. It's very difficult to be in an industry that is not only always changing, but always standing on the shoulders of giants that were there before you. So there have been places where I've worked where if you don't have the right degree, no one's going to listen to you. But at the same time, you keep self-promoting, having that confidence in not only knowing what your true value is, but also knowing what your skills are, but also owning where your weaknesses are too. If you go through constructive criticism with your year-end review with your boss saying, hey, you need to be a more empathetic leader, or you need to really dig down and understand what the lab needs, or you need to focus on whatever area to improve. Those are things where you have a weakness, and you don't want to necessarily acknowledge it right away sometimes, but those are the areas where you definitely need to focus on. And so those are the things that I've learned from the industry positions that I've been in. And then also something more important now than ever is understanding what a work-life balance is. My last position at the Andersons, I was on call 24/7 because the process was always running 365 days a year. It doesn't necessarily mean that's your life, but at the same time, it's hard to get away from a situation like that. Even if you're on vacation, your phone's always with you, and they'll call you if they need you. I loved interacting with the team that I had. I kind of got burnt out from it. So it was definitely time to make a change and know when to move on. Industry jobs tend to take up a lot of time. Not only are they necessarily harder to find the right fit for not only the individual, but at the same time, it's really important to understand when, as an example, I have currently an hour and 15-minute commute every day, and that's one way. So at the same time, I'm doing it, one, to save money right now to buy a house, but two, we're doing it mainly because I love what I do. And so it's just easy for the moment. I don't mind long drives, and it kind of helps me get ready for work and cool down from the day. So it's not a bad thing. But also, it's just making sure that you understand that industry jobs do take a lot of dedication. It's been, some people flourish in different settings, be it corporate R&D positions or corporate positions compared to production careers. I've had the benefit of being on both ends. I know what it's like to be in industry and R&D, but also be in industry and manufacturing. It's beneficial for me because now I see both perspectives, and I can use all that experience together where I'm at now to research for a purpose, and that's to make the process better or get something into the process that we never had before. So all these different features are really critical for what we do, and these are definite things that I hope all of you take away from this conversation. So at this time, I can open up the floor for any questions that you guys might have. Sure. Dr. Meager, looks like you have your hands up.
Just clapping.
Okay, sounds good.
Yeah, I'm really super impressed. I mean, I noticed you're using Jump. Was that because of your influence from me, or did you have that for your stats?
So when I was at Western, we actually learned stats through the program R, which is used a lot in multiple different settings. It's nice because it's open source, right? But I use Jump because every industry setting I've been in has used it. And so the features I like about it is you don't have to be a super genius in statistics to know exactly what to look for. But at the same time, the benefit to me and what I think about it is you have the drag and drop features so you can make figures relatively easily. And I like it because the figures are actually very presentable. I've also worked with Minitab before, and Minitab is really a strong, robust program system. A lot of engineers are trained on it. You just need to know the language a lot more in depth, I think, to use that kind of a software. Compared to something like R, where I felt like you need to be a computer programmer to use it, and I'm not nearly that good with computers, I think. But Jump is just an awesome system. I really like it. And plus, if you have a license through Jump, you can go through free training sessions that they have regionally, which are pretty cool, usually held at West End hotels, which are always very fancy, which is kind of a nice thing. But also, they have the best support and best customer service that I've dealt with when it comes to statistics software too. So very, very good program.
I'll just add another comment, and I'm sure Dr. Meager probably agree, but it's amazing to see, because we've known you for a long time, and we knew you as an undergraduate, really
Sure.
look behind your ears, and now to see you surpass myself, and I guess I'll speak for myself, at least in this arena, that's really impressive to me.
Well, thank you. I appreciate that for sure. Yeah, and that goes to say I would not be here without you guys, for sure. Western was so foundational in not only in my education, but also in, getting into industry. All the professors that I interacted with, I still see a lot of their influences in what I do. Not only in the way I kind of critically think about things, but also are able to critique my own ideas. And so managing kind of my own personal expectations with what I'm trying to do in research, but also be able to critically think about the process, just sit back and reflect, okay, where do we go? What are we doing? What are we trying to achieve? So that's definitely been foundational in everything that I do still to this day.
I have tons of questions, but go ahead, Dr. Meager.
Do you miss academics? I mean, what's like the business life versus the academic life? Because I love it here, right? The schedule.
Right.
So yeah, how do you?
Yeah, well, the one thing I will say is in academia, you have a little bit more freedom with what you get to research. So that the creative side gets to come out a little bit more. You're not nearly as restricted in terms of what your focus can be. So that's something that I'm always going to miss, to have that creative, kind of creative freedom there. But at the same time, I love researching for a purpose. And so that's really, and I don't want to say academic research does not have a purpose. The field that you focus on when you get into industry is a lot more defined. And so, I've heard it from one of my mentors. They say, we're going to basically keep you between the ditches.
You want to study? Yeah, I'll go in the room.
Okay, thank you. You want to study?
Yeah.
You may. I had a quick question.
Sure.
So I've seen pharmaceutical companies and a lot of other more business-based places. They have a tendency to shelf, certain ideas because it threatens their bottom line and their ability to make money.
Sure.
And you were talking about, don't fall in love with a project, don't get too attached, But I mean, if it's something that's life-saving and it gets shelved, I mean, what can you do in that case?
You basically lobby to keep working on it. You might not see a benefit to it right away, and that company is going to focus both on short-term goals and long-term gains. And a lot of times what the focus is always on self-preservation when it comes to where you stand in the market years from now. So it might not be something that they see value in right away because it's going to be something that affects something 20 years out. And so when it comes to that far out, a lot of times it's not as predictable. And so one thing I've had projects that I've absolutely loved that got shelved on me. But what you do is you try and lobby for them again and say, okay, this year it's not a primary focus. Let's see what the focus looks like next year. Let's see what the budgeting looks like for next year, and just, bring the ideas back up. And so a lot of times, even though those projects are completely shelved or corporate doesn't necessarily want you to focus on it, it's not something that can't come back. Now, having not been directly in the pharmaceutical industry, I can't speak to that perspective nearly as much. I know Jason Tudor, who will be talking to you guys next week, he spent 5 years in biopharma, so he can probably speak a little bit more specifically to that than I could. But in the agriculture, crop protection or bioconversions field, it's definitely something that, eventually it will circle back around. 6 or 7 years ago, people weren't thinking about necessarily animal replacement products. But now obviously we have companies like Impossible Foods and Beyond brands that are completely vegan or completely vegetarian. And that's really a forefront that, we're focusing on now as an industry as a whole.
I think, when I think about it though, some projects that are like game changing, but the problem is that like they're projects that may fix everything, but they get shelved because then the company's not making money on it. Like, for example, say you come up with the perfect pesticide that doesn't affect humans or animals in any way, and it doesn't involve GMOs because people tend to avoid GMOs. So, but then it gets shelved because maybe, I don't know, that means it's perfect, and then they can't sell more products because of it.
Right. Well, the other thing you have to think about too is the cost of production. It could be, a theoretical silver bullet, but the problem is your cost to scale and produce it might be prohibitive. So even though it is a good solution, it's not necessarily something that's industry ready because the technology that needs to be built around it is not feasible at the moment too. So that could be another reason why projects get shelved. And it is unfortunate, but things like that happen all the time.
Thank you.
You're welcome.
Okay, so thanks for your presentation.
Sure.
So I wanted to know that how do you, for instance, you start working today, how do you get into the work? Are you trained on what to do, or you just know what to do? Is there a training you go through?
Sure. No,
I was looking at
absolutely.
everything I don't really understand. So is there a training you go through to know what to do, or you
Sure.
just get into the work?
Yeah, absolutely. So, some of the solutions that I've come up with for either production or for R&D purposes, that it's all from learned experience, right? So, if you're not comfortable in an area or it's something completely brand new and someone else knows how to do it, more companies are more than willing to interact with you and train you to get you into those spaces. So, it's definitely not something that you necessarily know all the time, especially being where I'm at right now. I'm more of the front end, kind of fuzzy tech discovery kind of side. Sometimes you're going based on the only publication that ever came out about this one particular field for this one particular compound that came back out in the 80s, or even in earlier than that. And it was the only time it was published, and you need to get this to manufacturing. So how do you do it? Well, we're going to try a bunch of different things and see if one of them hits. That's really what it is. A lot of times it can be trial and error. If it's something completely new and novel, then, it's really up to you to develop it on your own. And no one's going to just throw you into the deep end and say, hey, good luck. A lot of times what you will see is people will be more than happy to work with you to get you trained in as close as you can be to an area expert. And that's really what they want to try and focus on, is to make sure that you're comfortable doing what you need to do, as well as making sure that you're able to get into whatever stance you need to be comfortable working in a certain area.
Okay, thank you.
You're welcome. See ya.
Jeff,
Yep.
First of all, what would be if somebody wants an internship, how would they go about it? And secondly, can you kind of give a pay scale of some sort from beginner starting to
Sure.
what's the typical trajectory? And then the other thing, let me add one more, is how
Sure.
stressful is it? Because you get this idea, to me, is that industry is always this, you better be running as hard as you can, as fast as you can. And how do you do the work-life balance if it really is that way? But
Sure. Yeah, absolutely. So I'll go in order of the questions you asked. So for internships and things of that nature, internships are invaluable. They help you get your foot in the door for sure. Really, to find them, I would find either a job forum or something on, LinkedIn is a really good example of a website to find things like internships, because you can go to their job page and type in internship in whatever field. And a lot of times you'll get some hits back. There are other search engines like Indeed, or I'm not sure if anyone uses Career Builder anymore, but that's another one that could possibly work. But any one of those job posting forums or job search engines can help you find internships relatively close to the area you want to be. If you have a particular company in mind, you can also look on their careers page. I know companies like ADM and Kraft always tend to have yearly internships. A lot of times they can be co-op setups, which work where you go to school for a semester, then you go into a internship for a semester, and kind of continuously cycle like that. You see that a lot with engineering backgrounds and degree programs, which is pretty neat. But yeah, if you have an idea or a specific company in mind, or a specific field you want to get into, definitely research the companies that are big players in that area and see if they have internships available. A lot of times when it comes to internships, a lot of people will get that internship, they'll go finish their degree, go to grad school, work in the other job areas, and a lot of times they come back to that original internship that they had. I know my current manager, she started out at Kraft as an intern, went to grad school, and then came back, and she's been here since. So that's an example of what you can do for that. Pay scale wise, it's highly dependent upon company and degree that you have. Various different levels. It really depends on also the kind of products that you're making. Your salaries are always going to be higher if you're in a medically relevant field. So if you work at a company like Pfizer or Moderna or something of that nature, they tend to pay better throughout the entire listing of their positions, starting anywhere from, I'd say 45 to 60 a year. Relatively average, I would say, depending on what position you have. And then when you work up the ladder, where I'm finally at, which is principal scientist, your pay scale is significantly higher than that. Generally, hopefully in the six-figure range, for sure. It also, again, that's dependent upon area that you're in too. So if you're located in a major city, you tend to get paid on a higher scale, but that's also on the commodity of, living expenses generally tend to be higher there. And it's also industry dependent too, like I had mentioned. I know, as an example, manufacturing jobs tend to pay significantly better, but they also tend to involve significantly more work. And going back to your last question, which is being in industry, is it always a continuous push to get something out the door or get something scaled up? Yeah, that is a definite tenet to the company that you're with and the job that you're doing. If you're in manufacturing, you either make it or you don't, and you either pay your bills or you don't. And that's really how it is. It's feast or famine when it comes to manufacturing. A lot of times, one day's worth of outages can, culminate in almost millions of dollars of loss, depending on how much you produce on a daily basis, right? So the ethanol plant I used to work with, As an example, a plant of that size can produce up to 400 to 420,000 gallons of ethanol a day. If you lose one fermenter in that process, you lose a third of your production. And so it's really critical to maintain operations in a consistent, continuous basis. But that being said, if you have a very consistent process, it's not nearly as stressful. That's what it is. When it comes to research in the industry world or the corporate world, if you will, it's dependent upon area. Everybody has goals and deliverables they're supposed to make on certain projects that are expected to be met, but generally those are by the end of the year. And so it's not necessarily all or nothing, but at the same time, you have to make progress in what you do. That being said, you don't research for the sake of just doing research in academia. You're researching to be, making those publications or getting to graduate, coming to the conclusion of your project, because you can never start and stop something and already have your answer, right? You always want to make that progress to continuously improve upon what you know or know it better. And so that's really, I would say it's relational, but it's definitely higher stress being in as an industry scientist, for sure.
I've found a lot of people in academia are stressed too, just trying to get published though.
Yeah, I mean, that's an incredibly stressful thing. I've been published 5 times. I know every single time I'm waiting every single day, checking constantly to see if there's any feedback and what to work on next and what to revise. A lot of times when it comes to publications, you can get a comment back that says, you might need to think about doing this extra couple of experiments to really support your work. And, that could take, several days or weeks or months to actually get that data back and resolved. So that's definitely stressful, for sure. I completely
also
agree.
the pressure to publish as well.
Yeah, but I would also caution that statement that your institution drives that focus, right? So if you're at like a Research One university, like a University of Kentucky or University of Arkansas for Dr. Musser, you're expected to publish. It's publish or perish, right? I think that's the phrase that you always hear. Not necessarily, some institutions might have a number requirement for you to publish every year, or number of publications need to come out of your department per year or something of that nature. But, there's a lot of stress there too, but that could be university dependent as well.
The problem is I found that universities have a tendency to hire people more for publication than for teaching. In my experience, like they will hire a professor because they can do really good research, even though they can't teach.
Yeah.
And it's a problem, I think, with academia.
It can be. Yeah. There's things like that in the industry too. You might be the, original founder of a certain technique or technology that's used in industry, but you come in and you don't develop past that school of thought, and you don't necessarily change the field anymore. I've seen or heard of that kind of thing happening before too. But it goes back to the statement I made earlier about being adaptable. If you can make sure that you're always publishing or looking in different fields, but also being able to communicate that message, that skill of teaching or communicating is definitely can be a lost art. And so I totally understand that. But yeah, you can see it in multiple industries too, for sure. Any other questions?
I want to ask.
Sure.
Yes. Good afternoon, sir, and thanks for your presentation. You gave a wonderful one.
Thank you.
I want to ask 2 questions.
Sure.
Yeah. The first question is, I am aware of universities outside US. I don't know. I'm not sure of that in US. But some universities, or let me say almost all universities outside the US, promote their staff using the number of years they have used in the system and the number of publications they have. So that's the yardstick. So, but is it the same? No, it can't be the same. I don't know. But what's the yardstick for promotion of staff for promotion of staff that works in the industry. I want to know the yardstick. What do they look at before a staff is being promoted in the industry? Then, secondly, I know there may be sometimes a worker in the industry may have to do an overtime, not his fault. Probably there's a lot of project in his hands, and he resulted into doing some overtime jobs. Will he be paid for it, or is it industry related, or do industries pay workers when they do overtime jobs?
Sure. Yeah. So to go to your first question, what's the measuring stick or the yardstick for, getting promotions? Is it similar to academia? It can be. If you're in an R&D setting, in 1 year you produce 5 patents. That's a phenomenal thing. You will definitely get some promotions likely coming your way. You might become a technology platform leader, as an example, or something of that nature. Those things definitely come in time. What you want to find is a company that works on a meritocracy kind of system. If you're continuously improving, or bringing new ideas or making things get to manufacturing level, those definitely pay dividends in terms of your job promotion scalability, for sure. If you work at a company long enough and you've done a lot for that company, you will make it very far in that company. That being said, when it comes to those kinds of things, sometimes that company is not necessarily willing to pay back on that. And so that's when you have to make a change. I've done that on a couple different instances where I knew it felt like I was being undervalued. Whether or not I was, that was just my perception on it. And so I made changes. And I've, every time I've changed positions with different companies that I've been at, I've always improved my standing in terms of what scale I've gotten to. So I started out as, scientist 1, which is the lowest you can be in terms of a scientist position at ADM, made it to scientist 2, transitioned into more of a specialty field in fermentation engineer at Corteva Agriscience, moved to the Andersons. I was in management and managing a process. And now I'm a principal scientist, which is a top level science position at the company I'm at now. So I always continue to improve that way. And you can do that. I mean, sometimes it requires you to move between jobs or between fields to get those promotions that you think that you need for your career. To answer your second question, if you work overtime, do you get paid for it? If your salary, absolutely not. I'm just going to go out like it is. A lot of times, especially in the manufacturing industry I was in before being here at Kraft, I was putting in 60 to 70 hour weeks on average. I got paid for 40. That's just what the job entails. I was dedicated to those plants. It shows in the work that we've been able to do. But at the same time, you're paid for 40 hours, and that's just what it is. Unless you're hourly, then at that point, you will get overtime if you work over the number of hours required for earning overtime. And generally speaking, that's usually something like time and a half for working more than 40 hours a week. So I hope that answers your question.
Yeah, thank you.
You're welcome.
Mr. Noland, I'm curious.
Yep. Oh, I think you went back on mute.
I'm sorry. How do you feel the opportunities were as a master's graduate versus a PhD graduate, and how might that have been different? Pros and cons.
Sure. Yeah. I honestly think most job descriptions nowadays, especially for science-related positions, they'll be, as an example in my area, it'll be stated as fermentation scientist. And a lot of times they'll be written in that where they can recruit across all those different degree backgrounds. So if you have a bachelor's degree with X number of years of experience, master's degree with X number of years of experience, or a PhD with X number of years of experience. So a lot of times they write them like that so they can be somewhat specific, but also be ambiguous at the same time. So if you have like as an example, like I do 2 master's degrees, that gives me more of a purview of more experience in that I have multiple graduate degree backgrounds to hire into some of those positions. Now, in terms of being able to find positions compared to the difference between being a master's graduate or a PhD graduate, it really depends on the area you're looking into, also the timing of what you're looking into. I haven't kept up on it since I left academia, but I still don't know if there's an overproduction of PhDs. But only, I still think a select few, no matter what, will be the ones that end up like a Dr. Musser, where they're able to graduate with the PhD, go through their postdoc, and land a university position as a faculty member. Same thing in industry. I think it's very similar where, you go through school, you work at a position and get a job based on timing, where the company's hiring very frequently, and you can stay there for your entire career. So I mean, it really depends on where you're looking. I think there's equal chances across all degree ranges, whether or not you have a bachelor's, master's, or PhD. Sometimes having a PhD is not necessarily the be-all, end-all when it comes to an industry job, but having the experience of knowing how to do something in industry definitely pays dividends. I know certain positions that I've looked into. I know I've had job offers where I've outcompeted people with PhDs and MBAs and MDs even for that matter. But the company decided to select me for the hiring process, which is, it demonstrates that not only can you work with the people that you're going to be working with and communicate with them, but you also have the skills that they desire. So it's really dependent upon where you go and what you know, realistically.
Thank you.
Mm-hmm.
Do you ever have any problems working with people that may not know as much of the subject as you do? Like if there's infrastructural issues?
Sure. Yeah. So being able to work with a diverse background of people is always beneficial for you. Being in academia, you're always thrust into a diverse background of people, which is awesome. I think that helps you not only take something very scientifically complex as an example and consolidate it down into these are the 3 or 4 bullet points we need to know for doing what we need to do. But being able to communicate across people who know it as well as I do and people who don't really know it as well as I do. To me, I don't have a problem talking to anybody. I feel like there is communication issues with certain people, and that's just really a stylized communication process. So knowing how somebody responds to saying certain words or knowing how to necessarily say, okay, this is our big project. We're going to look at particle size. That is a material science background where we need to look at laser diffraction of particles in a fluid media. Or you can just say, we're going to put, this sample into the machine, run it, and we need to know what size the particles are in the fluid. It's realistically what the difference is being able to communicate that very complex message in a simple way to get the answers that you're looking for and to work with the people that you need to work with. So it can be somewhat difficult depending on who you're interacting with. I definitely feel like there are times where people know things way better than I do, and I feel like I'm asking, stupid questions like, okay, so why do we need to have oil in the system? And so that's something that, okay, it shows that I don't necessarily know, as an example, for that specific reason, why something is incorporated in it. But I ask the questions in a specific way of being able to ask for clarity on it. So being able to communicate a scientific or complex message back in a simple way helps no matter who you're talking to. So I feel like that is definitely beneficial. And so that's the easiest way to communicate with people.
Does anybody else have some question?
Yeah, generally, I just want to appreciate you for what you do.
Sure.
Because it's kind of inspiring and really motivating what you do to to aspire young scientists like us to be able to dream and believe in whatever we want to become and achieve in our academic pursuit. So I'm just going to take this minute to say thank you and appreciate you for what you do.
Absolutely. Yeah, I mean, I was very much like you all were, not 10 years ago. I mean, I was sitting in those same seats listening to Dr. Musser give me lectures on, what caterpillar spit does to tobacco plants, right? So, I mean, I was very, very much like you all, and I understand exactly where you are. And, I never thought I'd be where I was today, honestly, but, knowing that the foundation that I got at Western was incredible for what I do today. And so definitely value the things that you learn here, but also use it to your advantage. Definitely leverage what you learn every day, for sure. So I very much appreciate that. Thank you.
All right.
I have one other question to ask,
Sure.
and it may be related to a second question.
Sure.
Entering the industry, starting a career in an industry, or let me say, like entering the entry level in the industry. Does it relate to the certificate someone is having? Probably, for example, an entry level may need a BSc. Now, if somebody is having a PhD and wants to enter an industry via the entry level, will this discourage an employer from employing him, or does it depend on experience, or let me say, starting as an entry level? Does it depend on experience, or it depends on the certificate? So, can a PhD holder start as an entry level in an industry?
Sure. Yeah, to answer that question, yes. I mean, it depends on experience. It also depends on your educational background too. Yeah. So, me starting at ADM, I started in a Scientist 1 position with 2 master's degrees. That's typically a bachelor's level required position. So, stating that, it was, I don't necessarily say it was below me to start there, but I had never been in industry before. And so you definitely need to get your experience somewhere. So, yeah, a lot of times if you don't have the industry experience and you apply for a job with an educational background of, it could be higher than a bachelor's degree or not. Those are things that every employer takes into consideration. So, yeah, I would definitely say it's a balance of both. Degree as well as experience level.
Okay, thank you. And one more question: does this discourage an employer? Does this discourage an employer because, with my experience outside the country, outside the US, an employer would want to employ someone with a PhD holder for a job that needs a BSc holder, because probably they are afraid of the payment, or they are afraid of some other things. So does this encourage or discourage employer in the US or in the industry in the US?
Sure. Yeah. I can't necessarily speak to that directly. I will say, if you have experience, period, or education, period, most companies are going to find the best people that they need to do the job that they want. So, whether or not it's from the US or not, I don't think that necessarily matters. It could be something that requires sponsorship from a company. I think that's more of a critical tenet rather than necessarily whether or not you were educated outside the United States or not. So if you require visa sponsorship, you have to look for a company that, 1, is willing to do that, but 2, willing to do it at not only an entry level, but also an advanced level too. And so a lot of companies are willing to do that. There are some select companies that don't necessarily have the ability to do that. So it really depends on who you look for and what you want to do. So, if I'm sure Dr. Musser can send out my email information or contact information. If you guys have any more specific questions related to that, I can help you kind of search out companies that are not only willing to do sponsorships, but also willing to interact with students that have learned abroad, for sure.
Okay, thank you.
You're welcome.
And to make it more clearer, what I'm trying to say is having a higher certificate and applying for a job with a lower certificate to one's level. Does it discourage an employer from employing such a person?
I don't think it
because
necessarily.
I learned. Okay.
Yeah, and so the reason why I say that is, the specific certificate might not necessarily matter to the company as long as they find the right person for the job. So that's really what it is. I mean, that's going to be company specific, but I don't think it discourages an employer from hiring that individual or not.
Okay, thank you.
You're welcome.
Well, I thought you did a great job, Jeff. Thank you so much.
Thank you. I appreciate it.
I don't know if we can give a round of applause, but thank you so much.
I appreciate it. Thank you very much. And like I said, if anyone has any other questions, Dr. Musser, feel free to share my email information with everybody. I am on LinkedIn. If you just search my name, you'll find me. I don't look any different from my LinkedIn photo, so I'm pretty easy to find. So feel free to search me out. I'd be more than happy to talk to you guys there too.
Perfect. All right. So, well, if it seems like we're done for tonight. Thank you so much, Jeff, for spending your time with us. And
sure.
Like I said, you've been, I was really grateful to have had you as a student in my lab. And
I'm definitely appreciative of it too.
Well, take care, man.
Thank you. Appreciate it. Thanks, everyone.
Jeff, did you need anything else? I'll visit with you more later, I guess. But
we could definitely visit later. But yeah, sure. I'm definitely. I don't need anything else right now. But if there's anything that I could do for you, just let me know for sure.
Sounds good. Thank you, sir. I'll
Thank
talk to
you.
you later. And then I'll rejoin you all at 6 o'clock. Sound good? So I'll rejoin you all at 6 o'clock, and we'll go for about 30 or 40 minutes.
All right, Doctor.
Sounds good.
Okay.
Sounds good. See you all.